Okay, welcome everybody to our webinar for the Columbia University Northern Plain Super Fund Research Program, our monthly webinar. We are very excited today to have with us Dr. Allison Kupsco, who is going to be sharing with us some of her experience and work learning about how epigenetics can help us to better characterize environmentally related disease in Indigenous communities. And Allison is an assistant professor in the Department of Environmental Health Sciences at Columbia University. She's been very heavily involved with the Superfund research program, with the development, thinking of the ideas for the research that we are doing. And she's involved in primarily in Project 3, which is a project that is an epidemiological study, but also she's going to be involved also in the Project 4, which is a more experimental study. So with this introduction, I hope we have now a good audience. So wonderful, Allison, we are looking forward to your presentation. Oh, and you are muted, I think.
Yeah, thanks, Anna. Can you hear me?
Yeah, perfect.
Wonderful. Thank you. Okay, let me get this started.
And let me say, before you start, we can see the now perfect. I wanted to say, please ask questions. We want to make this very dynamic. Put your comments in the chat, and then Allison will be able to answer them at the end. And we really want to make this very engaging. So feel free to put your questions as you have them.
Absolutely. Sorry about that. My screen was doing something a little bit weird. Let me try this again. We can try and start this over. Something weird with the okay, can you see that? Okay, great, thank you. Awesome, thanks so much, Anna, and thanks everyone for joining me today. Going to talk a little bit about epigenetics and what we can learn from epigenetics and Strong Heart and how this relates to our Superfund research program. I'm only going to talk for about a half an hour, so we'll have plenty of time for discussion and things like that afterwards. And I'm trying to keep this sort of as non-technical as possible so that it's accessible to everyone, but I'm absolutely happy to answer questions and so on as we go. And before I start, I think I really need to acknowledge a lot of people who have done the bulk of the work with epigenetics in the Superfund and Strong Heart study, particularly Anna, Arte, Shelley, and Maria, who have done an amazing amount of work in characterizing the epigenome and metals within Strong Heart, as well as the investigators and the participants that come from all of these institutions. This is really kind of an ongoing piece of work that I more recently have joined them, and I'm really excited to be a part of this team. So just to kind of give us a little bit of background, our Superfund research program is focused primarily on Indigenous health and health in American Indian communities. And most of us are aware by now that the burden of disease among these American Indian and Alaska Native communities is much higher than among their counterparts that are more Caucasian or white. So here I have the average age of mortality by disease or ethnicity, with the bars representing white individuals and the lines representing American Indian and Alaska Native individuals, and we can see that the mortality of ages from heart disease, lung cancer, COPD, diabetes, and stroke is much lower. And in fact, Native Americans suffer disproportionately from diabetes in comparison to all other race and ethnicities, and this is one of the focuses of my work. In addition to this kind of increased burden of disease, we see that Indigenous communities are exposed to a number of environmental factors that could influence their health. And this includes things such as proximity to mining, legacy mining throughout the Western United States. This map here that I'm showing overlays the Native American reservations, this crosshatch, with these abandoned mines, and we can see that there is a lot of overlap in the US. This is reflected by levels of hazardous metals and other pollutants present in drinking water. So I'm just showing here an example of uranium levels in public water systems that were measured throughout the Western United States with individuals with drinking water levels at higher concentrations in the darker colors in comparison to those with lower levels. And we can see that a lot of these areas, these also overlap with our Indigenous communities. And so a lot of our understanding of American Indian health and the kind of drivers of that has come from the Strong Heart study, which many of you may be familiar with by now. For those of you that are not, I'll just give a very quick background. The Strong Heart study is the largest study of American Indian cardiovascular disease in the United States. It started in 1989 with 13 tribes and communities across Arizona, Oklahoma, and North and South Dakota. And they originally recruited over 4,500 participants from this first study. This was expanded to include the family study of family members for original participants in the late 90s and early 2000s. And now we have about 3,700 or 3,000 individuals that have been enrolled. And this study is ongoing. And while we've learned a lot from the Strong Heart study about determinants of disease, one of the kind of, my opinion, in terms of our field, kind of more important findings is, the understanding of how exposure to heavy metals and our environment can impact health. And so what we see here is kind of the culmination of 15 years of work in Strong Heart in understanding how these different metals, such as arsenic, cadmium, zinc, and uranium, can influence health and disease. Particularly things like lung function, cardiovascular disease, including atherosclerosis and peripheral artery disease, cancers, diabetes, cognitive decline, and kidney disease. And all of these metals have been associated over the years with kind of long-term health in the Strong Heart study for many of these outcomes. And what's great and unique about Strong Heart is that because of the study design and the long-term nature of it, we really have the opportunity to investigate these kind of exposure and health relationships over time and how they persist and how they change. And so what I'm showing here is the design of the Strong Heart study from the first visit in 1989 to 91, and the visits thereafter that is, I think visit 7 is just finishing up now. And so we really have a great amount of data. They measured things like urinary metals at visit 1, subsets at visit 2 and 3. We have a large number of participants visit 4 and 5, which is over 10 years, 15 years after that original visit. And now the metals lab is just finishing up the metals measures at visit 7. And so we'll have a lot of amazing data on exposure to metals over time and how that changes with our environments, with different regulations and policies that have been put in place, and with changing technologies. But in addition to these metals exposures that we can measure over time in urine, we also have kind of biological measures that we can collect in blood. And this includes things like DNA methylation, which I'll talk about in a minute, and other biological responses. We have blood that's been collected at visit 1, at visit 4, at visit 5, and at visit 7 that we're able to measure these kind of biological responses over time. And this is able to help us answer a lot of questions regarding the pathogenesis of disease. So when I say DNA methylation of epigenetics measured in blood, what do I really mean? So we know that our genes and our genetic code is what determines who we are, how our bodies are put together, how they function, some of what diseases we develop, cancers, and so on. But what really controls whether this DNA and these genes are turned on and off is epigenetics. And so these are marks that are made on top of our DNA. They're added on to our normal genetic code without changing that underlying code. And these marks that are added on top can turn gene expression on and off, depending on what genes need to be expressed at different times in our lives. And so I think kind of one really elegant way of describing this is thinking about studying for a test, right, in school. If you're taking a class, you often have a textbook that you're given, and the textbook, it has a lot of information, but not all of that information might be relevant for your final exam that you're going to be thinking of. And so as you go through your lectures, you can read that text, which would be equivalent to the DNA sequence, and you can take notes, right? You can highlight things that you think are going to be important, or write down what the professor is saying that is important. And these can be thought of as our epigenetic marks. They're marks that are made on top of that underlying text that tells you, what you need to know for the actual exam. And then what we would call our phenotype would be the info that you actually remember for the test and that you put down. And so we have all of these sorts of layers that we can put together to help translate our genetic code into our health and disease. And one of the things that make DNA methylation or epigenetics kind of unique is that it can form an environmental fingerprint. So this DNA methylation fingerprint is modifiable, meaning that it can change in response to our environments or our health status. It's also stable, so it can persist these changes to our DNA methylation and the genes that might be turned on or off even after the exposure stops. One example of this is smoking, where there have been a number of studies that have been able to say, okay, even 20 years after you've stopped smoking, we can still see that you were a smoker in your action. And then the kind of last important point of DNA methylation is that it's heritable, which means that all of your cells, as they divide, might include it. And it's also heritable potentially to your next generation. That's what makes it really exciting. And so I have this diagram here, which shows kind of an overview of how our external environment might influence our epigenetics and our epigenome, which then might influence our later health disease. And I kind of want to give just a quick visual. I really like this visual of what this might look like. So the factors that we know that can modify our DNA methylation include things like disease, aging, smoking, chemotherapy, environmental pollutants, and so on. But this is a change that happens over time. So for example, what I'm showing here are two of our chromosomes, which is basically a compact version of all of our DNA put together, that are taken from twins. And twins, we know, have the exact same underlying genetic code. And so they should be equivalent. And what they've done here is they've labeled the epigenome and the DNA methylation in red for one twin and in green for another twin, and then overlaid them on top. And in this particular case, when we're talking about light, red and green make yellow. And so what we can see is that in, oopsie, not that far, in twins that are, only 3 years old, there's a lot of yellow here. And that's because the green and red and the epigenome for these two individuals overlaps a lot. But by the time they get to 50 years old, we can see that there's a lot of difference here. We have a lot of distinct green marks, and we have a lot of distinct red marks. And this shows us that even though these two twins have the same DNA, their two separate experiences have led to changes over time that change their gene expression, change their health, and change their exposure. And so how can we use DNA methylation and epigenetics to inform our environmental health in the strong heart study? The way that I can think of this is kind of 3 broad concepts. The first is that it helps us understand, the biology underlying environmental health disease, right? What is happening in our bodies from these environmental exposures that lead to this health changes. But other ways that it can be used are to identify past exposures. So because it's something that can be stable over time, maybe we can look into our past exposures using our epigenome. And then the third way is to potentially predict future risk of disease. And so I'm going to give you kind of a quick overview or an example of a few studies that we've done or that have been done in Strong Heart that can kind of get at each one of these factors. So first, in terms of biological understanding, I really want to highlight all of the great work that has been done in Strong Heart in terms of epigenetics. disease. There have been a lot of work showing that epigenetics is related to diabetes, related to cardiovascular disease, related to lung disease, and so on. And I just want to highlight this one study that they published just a couple of years ago, looking at arsenic levels and how it might relate to cardiovascular disease and diabetes. And what Arthe was able to show in collaboration with a number of others is that changes in DNA methylation that were related to arsenic were also related to cardiovascular disease. And these are changes that were not only identified within Strong Heart, but within a number of other cohorts that, you know, we can say, oh, this is consistent. And so maybe these changes in DNA methylation might actually be underlying the arsenic-induced cardiovascular diseases. And importantly, collaborators, Corin Mann, who's also involved with the Superfund, was able to replicate a number of these sites, these epigenetic changes in mice. And so, you know, we can say, oh, this is something that we see in animals, this is something that we see in humans, this is something that we see across populations. So maybe these are actual mechanisms underlying this disease. And so this is a really great work that was done, hoping, you know, we'll have a lot more of this to come. I want to share with you a little bit of the work that I have done in collaboration with the Strong Heart Study and some of our trainees, particularly focusing on biomarkers of exposure. And so, you know, oftentimes in terms of research, we're challenged by things like recall errors or other practical constraints. You know, not everything that we're exposed to throughout our lives can easily be measured, right? Some of things are data that we didn't get to collect, right? Like we thought, oh, maybe, you know, wish we could have done this, but it's too late now. Otherwise, it might be data that's hard to collect, such as information on things like childhood trauma, which is very difficult to define, or data that's prone to error, things that people maybe don't remember very well, like your diet over the last year, right? I don't remember what I ate yesterday, much less, you know, 6 months ago. And so, or things that people are more likely to misreport, right? Like illegal substance use. And so can we use our epigenome to measure these things that might be difficult to measure in other ways? And one way that we've applied that so far in Strong Heart are with epigenetic biomarkers for lead exposure. So you may be familiar that lead is an extremely toxic metal. It is particularly harmful to young children, as well as to adults and pregnant women. It causes a number of neurological changes, but it's also been shown to be related to kidney disease and cardiovascular changes. And the gold standard for measuring exposure in our lives is actually in our bone. And so there are X-ray measures that we can use to, in our legs, in our tibias and our patella, to measure chronic lead exposure that's occurred really over time. But this is actually rather expensive to do. It's difficult to do, and it's something that you really have to plan for in advance. And so there are actually not that many cohorts that have measured lead in bone. And so one of our collaborators, Dr. Elena Colicino, who's at Mount Sinai now, asked the question is, can we use DNA methylation measured in blood to approximate what our lead exposures would be in bone? And so what she did is she measured DNA methylation through the genome and said, okay, can we use a combination of DNA methylation changes and estimate patella and tibia lead exposure? And these are some correlation plots showing that, you know, the correlation between the actual lead levels in the patella and the estimated lead levels from this DNA methylation was about 0.6. And these are kind of moderate correlations. And so one of the trainees that we have working with the Superfund, who's been working with myself and with Anna for the last couple of years, Will Lieberman-Cribben, did this work applying this lead biomarker to the Strong Heart Study and saying, okay, can we use this lead biomarker that was developed in another population to predict lead exposure in the patella and in the tibia from Strong Heart? And are these lead biomarkers related to cardiovascular disease? And so what he looked at was DNA methylation measured at visit 1, as well as cardiovascular disease mortality through, I think it was 2019, and cardiovascular disease incidence, and found that greater tibia lead, as measured through this epigenetic biomarker, was associated with greater cardiovascular disease mortality overall, suggesting that this kind of lead biomarker from epigenetics is actually telling us something about our past lead exposures. Interestingly, I'm showing this here. It's kind of a lot to look at, so don't worry too much about all of the individual points. But in subgroup analyses where we said, okay, is this an association between lead and cardiovascular disease mortality stronger in individuals that smoked, for example, in individuals that were part of tribes located in Arizona and Oklahoma or Dakota, and those types of things. And what he found was actually the association was much stronger in men in comparison to women. And our question is this a biological effect or is it a technical effect? And I think that this is kind of highlighting a lot of what we need to learn about epigenetic biomarkers of exposure, because the original biomarker was developed in a male population. And so we don't have a ton of information on women, and that's something that we'll need to add over time. And so it might not be that the impact of lead was actually greater in men than women, right? We might not be able to make that conclusion, but it might be that the biomarker is performing better in men. And so this is something that we will eventually need to follow up on. But overall, this is really an indicator that in strong heart that these epigenetic biomarkers of exposure may be able to tell us something about our past exposures that we have not been able to measure. The next sort of topic and way that we can use epigenetics is estimating our health and our health status. And one way that this has been sort of operationalized in the field is the concept of biological aging. And so we know that things like I'm calling health span and lifespan are really different between individuals, right? Someone might live to 95, whereas someone else may pass away at the age of 80, right? Or someone might develop cardiovascular disease very early in life, whereas someone else might be healthy up until the day that they died. And thus this kind of calendar age based on the days that you've been alive might not be an accurate indicator of your actual health status and your actual age. And so that's where this sort of idea of biological aging came about. And so in order to kind of estimate our biological age in individuals, there have been a number of studies that have estimated what are called epigenetic clocks. And what these are age estimates based on our epigenome rather than based on our calendar age. And so I'm highlighting 3 of them here that have been developed more recently. They're called pheno-age, rim-age, and Dunedin pace. And these are sort of developed based on our aging-related processes. So for example, pheno-age is a predictor of aging phenotypes. It's been trained on variables that are associated with our health span over time. In contrast, rim-age is much more related to mortality. It was intended to predict lifespan. So it's trained on proteins that are predictive of mortality. And then finally, the Dunedin pace clock is a little bit different, and it's trained on a rate. So how do our biomarkers of aging change over time? And these 3 clocks all sort of function on the concept of epigenetic age acceleration, whereby if your chronological age, so if you are 65 years old exactly, is exactly equivalent to your epigenetic age at 65, then you don't have any age acceleration. But let's say your epigenetic age is greater than your chronological age, then perhaps you have positive epigenetic age acceleration. Your biological age is greater than your chronological. In contrast, if your epigenetic age is less than your chronological age, then perhaps you have negative epigenetic age acceleration, and you might be healthier. And so how can we implement this to gain insight into environmental exposures in strong heart? Well, we had a really talented master's student who worked with us, Kayla Boyer, who was interested in answering this question. And she asked if we were able to determine the impact of individual metals that were measured at visit 1 in strong heart on epigenetic aging, as well as determining the impact of the total metal mixture on epigenetic aging. So is it individual metals that might be involved, or is it the total metal exposure that might be important? And what she found is there's a lot of text on this page. Don't worry about it unless you're interested in the very specifics. But what she found was that cadmium and zinc were associated with increased epigenetic aging in the strong heart study. So what we're looking at here are effect estimates from linear models examining quartiles of metals exposure and these three epigenetic clocks, the Dunedin pace clock, the Grimm age clock, and the pheno age clock. And in comparison to lower metals exposure, so metals less than the 25th percentile, individuals that had higher metals exposure had greater epigenetic age acceleration, particularly for cadmium, but also the sort of nonlinear association for zinc as well. And these are in our kind of fully adjusted models, adjusted for a number of important confounders. When we use mixture modeling, so in this instance, we used a method called Bayesian kernel machine regression, which I'm not going to explain too much about, but basically we see these exposure response curves where we have increasing levels of metal concentrations and the changes in our Dunedin pace outcome, our Grimm age outcome, and our pheno age outcome. And we can see that there's a very strong relationship between increasing cadmium levels and changes in our epigenetic age acceleration. We see a small amount of that for zinc, but not as much. And then we see a sort of nonlinear relationship with selenium as well. And so we have all of these metals. We have arsenic, cadmium, we have tungsten, zinc, selenium, and molybdenum. And this is a mixture that is a complex mixture. So we're exposed to all of these metals simultaneously, not just one at a time. And so what we need to do is consider them all together. And these metals can actually be divided up by essential status or non-essential status. So things like zinc, selenium, and molybdenum are essential metals for our health. We need them in order to survive. But non-essential metals, such as arsenic, cadmium, and tungsten, we don't need, right? These are heavy metals that may only act negatively. And so what we did was we looked at the impact of the total metal mixture overall, which is this kind of gray row here, and then separated by essential versus non-essential. And what we can see is that particularly for Grimm age, as we increase our metals quantile, we have greater impact on epigenetic age acceleration for our non-essential metals. But when we look at our essential metals, we actually see the opposite effect. And so what this suggests is that for these non-essential metals might be negative influencing our aging and our aging-related processes, where essential metals might be improving them. And so this is something that we think is quite interesting and is going to be looked into further. I also want to share that we looked at whether these associations persisted in individuals that were smokers versus individuals that were non-smokers. We know that cadmium levels are very high in cigarettes, and so individuals who were current smokers may have very high levels of cadmium. But actually what we saw was that these associations were greater in individuals that never smoked in comparison to those that used to smoke, particularly for pheno age, which is our phenotypic aging indicator. And we see it a lot with zinc. And so what you're looking at here is for the red triangles, these are never smokers. They have a stronger association than our black circles, which are our ever smokers. And so from this particular study, what we're able to take away is that cadmium and zinc in urine might be related to biological processes that underlie aging-related diseases. And that essential metals might mitigate the impact of these non-essential metals, which is going to have a lot of implications for kind of future disease risk. And that understanding the combination of these is critical to advancing our disease. And so just to kind of wrap up the work that's been done in strong heart study is that epigenetics has really been able to provide insight into the mechanisms of disease, such as the study that I showed on arsenic and cardiovascular disease. It allows us to examine the impact of things like lead in the absence of measured bone values, and then to estimate the long-term impacts of these exposures on aging, including potential improvement with alternate exposures. And so this is kind of a lot of the foundation of where we are with our Superfund program, but we're excited to start taking our next steps. Our program focuses mostly on uranium and arsenic levels, which are two of our priority pollutants for communities in the Northern Plains area. So we're quite interested in them. We will be extending our DNA methylation analyses across about 15 years, which will allow us to see how these things are changing over time, which will be very important, allow us to assess the long-term impact of metals. And we'll also be adding metabolomics data, which will be more of a functional. So we're really excited to get started on that. So with that, there's a lot of people to acknowledge. I can't even list everyone here, but we have an amazing Superfund team that we've been working with, which I'm very fortunate to be a part of. I'm also listing the individuals that were involved with the papers that I spoke of. Of course, our funding sources, including the Superfund. And so with that, I'm happy to take any questions that people might have.
Thank you so much, Allison. And I can see that there are some questions already. I don't know if you want to remove the slides so that we can see the people. People can come on camera if they want. You can bring the slides if you need it.
Yeah, absolutely.
Wonderful, Jill. Yes.
Hi, hello, thank you. Can you hear me?
Yes.
Okay, great. I'm not feeling very well, so I'm a little under the weather, but I'm going to do my best here. My name is Jill Aquino. I am an environmental health nurse. I live in New Jersey, and I work with the Ramapo-Lenape Turtle Clan in Ringwood, New Jersey. I don't know if any of you are familiar. The Ford Motor Company in the 60s and 70s dumped their toxins on the Ramapo. And sadly, the tribe is dying of very rare cancers at alarming rates, even though it's 2024, you know, the toxics have impacted the community greatly. My question, Allison, and thank you for this wonderful presentation. As an environmental health nurse, what we're doing is we're assessing tribal members' urine and blood for toxic metals. And what we're finding is when we receive the results back, the tribe does not want to know, the tribal members who have volunteered to have their blood and urine collected, they don't want to know the results. And so is that just a common theme in marginalized communities who have suffered as the Ramapo have? Is that something that's unique to the Ramapo? Is that something that you see in your practice? And is that something that we as researchers, nurses, scientists, et cetera, we just accept that this is what their asks are, they do not want to know, and we are just respectful of their wishes? Your thoughts, please. Thank you.
That's a great point, Jill. I think there's a little bit of feedback, but definitely, I mean, extremely important. And I think Anna's probably better at answering that question than I would be, because report back is not my area of expertise, but I don't think we have the same feelings.
Maybe something that can be helpful, and I would love, I know that there are some people here who have a lot of experience, like Lyle Best, who has joined us, but something that I think I would like to start, this is a very important question and conversation, which can apply to reporting the metals data as well as other types of data. The Songhar study was built on a lot of trust and community engagement, like going along over many years, and maintaining that trust, which is not an easy job. And that means that the tribes are informed all along the way of all the measures and everything that is going to take place, because they are very involved in that process. I don't know, in your case example that you mentioned, if this is an issue of report back at the individual level, so reporting back to the individuals, or is this an issue of reporting back at the community level? So providing like summary reports and informing what the findings overall were. The Songhar study does a little bit of both. There is report back that go to the individuals, especially for the clinical assessment. So the clinical information is reported back. If we find something elevated a metal level that would be considered clinically relevant, that would be also be reported back. But for the ongoing participants. But most of the report back that we do is actually at the community level, which means that we meet with tribal partners, we do report. For instance, we've done that a lot with the water sampling that we've done. We have newsletters where we summarize the main findings. For instance, the epigenetic work that Allison has been presented, we provided Anne Bozak, who was another trainee involved with the Superfund Center. She prepared some really very helpful summaries that went through the newsletters that go back to the participants. We also do with tribal agencies like the natural resources, we work with them, and we've plan also like interventions related to those findings, because sometimes reporting back means, and so what? What do we do with that information?
Right.
So that I think, but always making sure that you are working along the way together so that there are not surprises with the data that was measured or how is that utilized. So I don't know if that's helpful.
Yes, and it's very helpful. And so I just want to say two things, and I know other people want to speak. I don't want to take up everyone's time here. So we have done individual report backs for those who want individual. We've only had one person so far say, yes, I want to know. Chief Man, who's the leader of the Turtle Clan, said he wants us to present to the community as an overall, this is the information based on the urine and blood findings with the metals. And so he wants us to educate his tribe, which we're happy to do, but we have to do it in a very sensitive manner because, you know, what we found is very alarming, of course. So thank you.
Thank you so much for sharing. This is the purpose of this webinar, so that we can learn from each other and find together the best way to do. I think there is another question in the chat. Yeah, Allison?
Yes, yeah, Laura, this is a great question. Look about the kind of results on the smoking and the effect modification that we're seeing by smoking status. This was surprising to us as well. The reason that we performed this analysis specifically is because we wanted to make sure that the results that we were finding weren't just due to smoking, right? That we weren't measuring the impact of cigarette smoking on aging, which we know is going to be a profound impact. And so we were also kind of quite surprised to find that the impact on the epigenome, the aging that we're measuring with the epigenome, right? We have to remember that this is a surrogate measure, not necessarily the aging itself, is stronger in these sort of never smokers. And we're still sort of trying to figure that out. I think we'll have a little bit more information. So right now, the data that we're looking at is cross-sectional. It's not longitudinal. So once we're able to have more longitudinal data, I think we'll be able to piece that out a little bit more. But we were also quite surprised with that finding. Are there any other questions? Happy to.
I'm sure there must be questions.
I have one. I'm Harpreet. I'm with ATSDR, and I'm just curious to know when you talked about epigenetic aging, and please know that this is not my area. So I'm asking this as a naive person here. So when you talk about epigenetic aging, and when you said that it could be a surrogate for aging, right? And with aging is associated with cancer risk. So can we actually say that the epigenetic aging is associated or could be associated with cancer risk? Do you see my point?
Yes. So cancer is definitely not my area of expertise. I do believe that there are studies that have looked at epigenetic age and cancer risk and have found found associations there. Epigenetic age biomarkers are that is something we should do in the Strong Heart study, because we have a lot of great information there, and I think it would be really helpful. The epigenetic age biomarkers are intended to be predictive, so there's no sort of causal connection between the exact DNA methylation changes that we measure with epigenetic age and, for example, cancer, but it might be something that you could say, okay, maybe there might be some cancer-related processes occurring early on.
Okay. Thank you so much.
Thanks.
That's helpful.
Danielle, did you have a question?
Hi, Allison. Yeah, thanks. That was really great talk. Kind of a loaded question, but it's also because of my naivete. What's going to take it to the point where epigenetic aging is actually going to be used when you go to the doctor and they're able to, you know, take all these tests on you and then determine your epigenetic age? I mean, I can only imagine, like with genetics, you know, if insurance companies hear about it, they'll say, oh, obviously your epigenetic age is way older than your actual chronological age. Therefore, we're going to raise your insurance rates. But I'm just wondering, if and how would there ever come a time where you'd actually take somebody's epigenetic age at the clinical level?
Yeah, this is a fabulous question. I actually talk a little bit about this in the class that I teach because it's so fascinating, this issue. Basically, there are already individuals who are trying to measure their epigenetic age on an individual basis and reduce it with lifestyle interventions. Whether those interventions be something like dietary or it be something more extreme, like, for example, I've heard of people doing like chemotherapy to reduce their epigenetic age. There's this man in San Francisco who's some tech person that I cannot remember the name of. Arthur, do you remember his name? I cannot remember his name. Who's measured all of these things on himself over time.
Yeah, I don't remember the name either, but there are several companies doing that, and I mean, investing billions of dollars on that. So
yeah, you can actually Google on Amazon epigenetic age or epigenetic clock and buy yourself a kit for a couple $100s and measure your epigenetic age. So this is something that has already made it to the free market, but the actual interpretation of it, I think we're not there yet. There's not been enough intervention studies. You know, we don't have the kind of clinical trials to tell us whether a reduction in your epigenetic age is actually going to change your long-term health status. Who knows, maybe one day that will be, you know, we'll be there, but I think we have a ways to go. You know, these biomarkers have been validated now as functional across different study populations, right? We're talking about a very unique population here with the Strong Heart study. They've been used around the world in different genetic backgrounds. They've shown to be effective, but what aging means for different populations may be different, right? So aging in the US is associated with certain diseases, but in, for example, more kind of subsistence societies, it might be different. And so I think we're a ways from getting there, but we're trying, and I'm sure insurance will happen.
And Allison, I am thinking, and it's so great to get all these comments and input that makes us help us do better work with the repeated measures. As you mentioned, we are going to be doing in the family, and some of the participants were both in the original cohort and the family. So we'll have some repeated DNA methylation. So we could potentially look at participants who have like improved their epigenetic aging, maybe. If there are repeated measures of and versus people who, or maybe people who accelerate more versus accelerate less, and that could be a very interesting metric to look at long-term health effects if we do this type of work, which is exciting that we can maybe do some of this with the new measurements that we are planning. That's exciting. I see there is another question from. Related to the metallothionein, which is also from Tim Silselsky, which is also very interesting. Did you see the question?
Yes, I see that. Tim is wondering if we have looked at metallothionein genotypes or expression levels. We do not have the samples preserved in a way that we have done the expression, not the gene expression. We don't have those. I suppose maybe we could look at protein expression in the future. Cost is always an issue there. But I do think we are actually completing a large amount of genotyping right now. And so we will have some great genotype and GWAS information available in the future. And so that's a great idea of something to look into.
And maybe, I don't know, Catherine, I think she's Catherine Schilling, I believe she is in the session. Maybe Catherine, do you want to share a little bit your ideas about the metallothionein? Catherine is doing a pilot study to measure metallothionein in MESA and potentially, and wave scan, which is another smaller cohort that we have. And potentially from that work, we could learn how relevant or how useful this is to measure metallothionein in urine, which, and that could also help. I am not sure.
Yes, Anna, you already summarized it so nicely. I don't have to say more. Yes.
Yeah, but Catherine thought this would be very important work to do because how important metallothionein is for cadmium metabolism, zinc metabolism. So yeah, thank you so much for that question. Lyle, can see. Yeah, thank you for joining, Lyle.
Yeah, thank you. I was just wondering.
We lost you.
We lost your. We could hear you at the beginning, and then we lost you.
Okay, I'll try again. When you look at changes in epigenomics over time, it makes the question come up: how accurate are the measurements to begin with? If you do the same sample over, you get good precision in measurement.
Yeah, the precision is actually pretty good, actually. For this particular assay, it's an array-based, microarray-based format, so it's a little bit more precise than something like sequencing. So we do get pretty decent precision, but we do see things like batch effects and kind of changes in that. So that's something that we always have to account for when we do these analyses. But overall, actually, the method is pretty well validated, which is great. Yeah, and I see we have some people who have put articles in the chat about Brian Johnson, I believe is his name. So thank you all for sharing those. These are absolutely fascinating. So I recommend you take a look. Oh, I see. Sorry, Wending has a question.
Yeah.
If we could talk about how to measure differential aging for different tissue types. Is there a way to measure that from blood samples? So most of the kind of epigenetic aging measures that have been developed were built specifically for blood. The original epigenetic aging clocks that were produced were supposed to be sort of pan tissue. And so they chose measures, they used a lot of publicly available data that allowed us to sort of normalize across many different tissues in the body from a large sample size. The majority of studies that are coming out now that are requiring more information on health status, mortality, change over time are built specifically for blood. And I think there is still an outstanding question about whether sort of other tissue types are going to behave the same for these types of measures over time. Depending on the goal of the study, right, it might be that blood is the optimal biomarker for you. However, if you're more interested in isolating rate of change from a particular tissue, then that's going to take a bit more kind of work and optimization and methods development, because that's not something that's been done as much. But there are others that have come out looking at aging in buccal cells and saliva, as well as other types of easily accessible biosamples for human population studies, because of course blood is the one that we have most frequently. I hope that answers your question.
Great. I have a question myself, Allison. I was wondering, it's very interesting to see that for the 3 epigenetic aging biomarkers that you showed, the number of CpGs is so different, from only 173 in the Dunedin pace, when it's an amazing predictor, like it works really well, to more than 1,000 in the Grimm age. And I was wondering, is there any overlap among the CpGs selected by these 3 biomarkers that you showed? And how can we do this very efficient, finding these very efficient biomarkers that maybe don't need so much information? I found that quite interesting.
Yeah, you're absolutely right. That is a wonderful question. The RIM age clock CPGs have not been published, the exact ones. So I don't know which ones those are. I believe there is not a lot of overlap between the clocks and the CPGs that are included. I think Arte probably knows that better than I do. Do you know how much overlap there is?
I don't know about the overlap, but again, as you said, we have no access to which CPGs are the ones that are composed, that are included in the GRIM age clock. So, but it will be interesting to look at that.
Yeah.
If possible.
There is not a lot of overlap. And actually, in my opinion, I think that 173 is an extremely small number. There are 28 million CPGs in the genome, right? And so, 173 CPGs, and the fact that it is so predictive is really interesting. And I think there's been, some work in trying to identify why changes in these CPGs are so predictive of health and aging and work in animals. But actually, what's interesting is a lot of the CPGs that are from these original clocks are not conserved in animal models. And so there's actually been a totally separate animal epigenetic aging clock that has been developed for that particular reason. And so it's a little bit tricky to understand, and that's why these predictive models can be sort of challenging, I would say.
Very good. Good. I think all of these questions and comments are extremely helpful, especially these questions about how is it modifiable and how, because I think that's what the communities are very interested. How can this information be used for interventions and real actions? So that's really, thank you, everybody. Any other questions, comments?
There's another one in the chat.
Oh, very good. Yes. So
yeah, sorry, go ahead, Ona.
No, I think, this is an environmental epidemiology study, or sometimes people call this molecular epidemiology or environmental and molecular epidemiology. So it's all observational, but the intervention aspects are quite important. And so the question is, which are for instance, if we remove one of the big questions we are trying to answer in the Superfund research program is what's the difference, what's the impact of removing the exposures? If you remove or what's the role of the past exposures, the long-term past exposures versus the current exposures? Do these past exposures have a mark? And or can we really prevent these processes, this biological aging, molecular pathways, epigenetic markers? Can they be modified? And is that relevant for disease processes? So we hope that removing environmental exposures matters. We've seen, this is some novel data that was just generated by our Trace Metals Lab, led by work by Catherine Schilling and Ronnie Gramonjat, who is here. I also saw the members of the team, Chugo, in our lab, who have been working on this data, Jagoda, who is not here today. And we've seen some trends, declining exposure, which is very important. Will Lieberman also run this analysis very recently. This is how we now know that exposures have declined. This is wonderful news for the communities. The fact that we knew we had high exposures and now the exposures are much lower, this is very important. And now the big question is this decline related? Was this a successful intervention? And is this related to better health? So we hope to answer that questions. But there are other interventions that might be relevant through nutrition, which is also a big element for us, for the Superfund project, both in our epidemiological work as well as in our animal experimental work. We really want to look at these potential through nutrition interventions, but there might be other interventions that we can think of. So I think with this, are there other questions or comments? I think we've covered a lot of ground, which is amazing. I want to thank Allison for this really excellent presentation. I think it's a role model that we should all learn from because it was very informative but very well done, very succinct at the same time. And I think this has allowed us to have a real discussion with lots of comments and ideas. And I think it's a real model for us to follow. For those of you who are interested, the presentation will be, want to listen again, the presentation and the discussion will actually be available through the YouTube channel for the Superfund Research Program at Columbia University. So thank you everybody for joining us today, and we'll follow up next month. I don't know, Whitney Collado or maybe Yoshi, if you want to tell us about the speaker next month in the last 2 minutes that we have. Because I believe it's going to be a very good presentation. I don't know, Yoshi, Whitney?
Yeah, sorry, I was trying to unmute. Yeah, so our guest speaker is going to be Stephanie Tucrow, who is a respected elder in the community. And I believe they will be talking about their work with medicinal plants and some of the teachings that they've been able to provide for both students that participated in the high school summer research program that Annie and I lead last summer, but also their teachings that they've been able to embed into the tribal colleges of the nation. So stay tuned for that one.
So stay tuned and see you next month, everybody. Thank you for joining. Bye-bye.